The second part of the cell life cycle after it goes through interphase is the division phase. Now, if we're talking about body cells here, like skin or muscle, these cells are going to divide through a process called mitosis. At the end of mitosis, it's going to make two identical cells. Reason why they're going to be identical, if you guys remember from the previous video, the DNA is going to be copied during the S phase of interphase. So each new cell is going to have the exact copy. We see the acronym to remember the phases is going to be PMAT. P stands for prophase, M metaphase, A anaphase, and T telophase, followed then by cytokinesis, which actually is going to split into our two identical daughter cells. Now, if we're just going to start with prophase, there are a couple key features that we can observe using a microscope that let us know the cell is getting ready for division. In prophase, we see that the nucleus is going to start to break down. So I haven't drawn a full nucleus here. I've drawn it kind of dashed because it's starting to disintegrate. Inside the nucleus, we see that the DNA, which when it's not dividing is called chromatin, it's stored loosely, it's going to start to condense around proteins called histones, and it's going to form chromosomes. In our body cells, we have 46 chromosomes. I don't have room to draw 46 chromosomes in this cell, so I'm only going to put 4 for the sake of keeping this video simple. So, nucleus breaks down. Chromosomes are actually going to become visible, and then we see that we have this little organelle next to the nucleus called centrioles are going to be here to help make spindle fibers in order to separate these chromosomes. So prophase, nucleus breaks down, chromosomes become visible, and the centrioles are located next to the nucleus. Now in metaphase, these centrioles are going to make spindle fibers that are going to pull the chromosomes to the middle of the cell. So metaphase is going to be middle. This is where we're going to see these chromosomes. And again, I started with 4, so I'm only going to put 4 chromosomes here in the middle of my cell. Now, the way that these chromosomes line up in the middle of the cell is we see these centrioles are going to be on opposite sides of the cell, and they have made these spindle fibers that attach to the centromere of these chromosomes. And they kind of do this little tug-of-war act to line them up in the middle. So let's go ahead and we can add our labels here. So my centrioles, I'm going to say labeled up here as well. And these are the spindle fibers that attach to the centromere of the chromosomes. In our next phase, anaphase, these chromosomes are going to be pulled apart. So ana is apart. We see that the tug-of-war action going on between these chromosomes are going to split it into the sister chromatids. We still will see that the centrioles have these spindle fibers. So we're going to go ahead and draw these here. But instead of seeing the full chromosome attached to it, we're going to see only half. And each of these halves are going to be identical. So this half right here, once we split it, is going to be identical to this half right here. So I have split these 4 chromosomes into their chromatids. Now, telophase, our next phase, we're getting close to dividing. The nucleus is going to start to reform. It's not fully formed just yet, so I'm not going to draw it as a solid line, but instead I'm going to draw it as a dashed line. These chromatids are going to start to relax and uncoil, and it's going to turn into chromatin, and it kind of looks like this cooked spaghetti. So it's unwinding and relaxing, and we see this happening in both locations of the cell. So let's go ahead and we'll add some labels here. Sorry for not labeling this. We still have our centrioles here. Put a label here and here. We see the spindle fibers. And then this is going to be a chromatid. So if we're going to add some labels to this picture, we see the nucleus is starting to form. And the DNA is going to relax and turn back into chromatin. Now, these are our phases for PMAT, so prophase, metaphase, anaphase, telophase. This is our period of division here. Now, to actually get to individual cells, we have to go through a process called cytokinesis. Cyto means cell, and kinesis we're going to cut. So we see that in the animal cell, this is going to start to pinch in until it forms two identical daughter cells. And these identical daughter cells are going to then have a fully formed nucleus. So I guess I can label my nucleus here as well, sorry. Nucleus is reforming. Here we have the nucleus is fully formed. And we see chromatin inside of these cells. And this is our process of mitosis. We have two identical daughter cells at the end.